Welcome everyone for being here today and listening to my little spiel. I got my master's at Youngstown State University in environmental science, so this is a lot about what I worked on for my master's. We were working on using stable hydrogen isotopes in order to look at change over a period of time. So one of the things that is really awesome about stable hydrogen isotopes in North America is that it deteriorates across the landscape in this really beautiful latitudinal pattern. Right? So it makes it really easy for assigning birds to approximate location. And we can do that because these isotopic ratios are actually fixed permanently into carotid tissues of birds. So there'll be a signature in feathers or perhaps in nails. And we can assess this in order to calculate the probability that an individual originated from a particular location. We can expand that then even farther to estimate the most likely region of origin for a population of individuals. So we specifically looked at the regional population of magnolia warblers in western Pennsylvania. So some of our goals for this project were to calculate tolerance intervals using these stable hydrogen signatures in order to visually represent the area of occupancy of the magnolia warbler within its breeding range. We then wanted to try and compare how the area of occupancy has changed over a period of time, and we wanted to be able to statistically quantify this change. And then from there, maybe assess why change is or is not occurring for this species. So again, we were looking at the magnolia warbler. In order to assess the current breeding latitudes, we pulled feathers from migrating magnolia warblers. We would pull feathers as they came through the Powder Mill Avian Research Center for banding. We then also pulled feathers from study skins at the Carnegie Museum of Natural History, and we had them analyzed in order to assess breeding latitudes in the past. So one of the first things we did after we had our feathers analyzed at the Cornell Isotope Lab was to georeference the breeding range for the magnolia warbler. So we kind of looked at the breeding range for the magnolia warbler and georeferenced it in ArcMap. And then we calculated tolerance intervals. So a tolerance interval is it's very similar to a confidence interval, except that instead of a confidence interval, this tolerance interval specifies a specific proportion of the population that falls within a range. So we computed tolerance intervals at the 50, 75, and 90% with a 95% confidence interval. So basically that just means we're looking at a range in which 50% of the population is most likely to fall with 95% confidence. We calculated these tolerance intervals using an R package. You can see the R package there. And they were all non-parametric. We didn't want to make any assumptions about the data that we collected. So these are what the tolerance intervals were. We had our 2013-2014 sample, and we ended up with two sample sets from the museum. There was a set of feathers that fell within 1895 to 1910, and a set of feathers that fell between 1963 and 1984. So you can see our sample sizes, and you can see our tolerance intervals there on the right, but these don't really mean a whole lot until you look at them visually. So this is what the tolerance intervals look like visually in ArcMap, right? You can see in the dark red, 50% of the population is going to fall within that dark red area. 75% of the population is going to fall within the lighter red, and then the pinkish color is an area that's going to hold about 90% of the magnolia warbler population. So once we have these tolerance intervals calculated for all of our sample sets, we can overlay them and kind of compare them. So when we're looking at them for visual comparison, they kind of look like this. This is a comparison between our 2013-2014 sample, and it's being compared to our oldest museum sample, the feathers that were between 1895 and 1910. So you can kind of see how they're different. You can see the darkest green is the 50% tolerance interval for the oldest feathers, and you can see the slanted lines is the 50% for the 2013-2014 sample. So you can kind of visually see how they're different. And we can do the same thing. This is our 2013-2014 sample compared to our intermediate museum set. So these were feathers between 1963 and 1984. Again, the change isn't quite as obvious, but you can still visually kind of see the change. An even better way to kind of show change over time is using standard deviation ellipses in ArcMap. So this is just for the 50% tolerance interval, with the blue one being our most recent feathers, the yellow is our intermediate, and the red is our oldest set of feathers. So correlation ellipse of the 50% tolerance interval, but you can kind of easily track the change over time. But we wanted to be able to statistically quantify these changes, and one of the ways we did that was through a bootstrap analysis. So in order to do this, we pooled all of my actual samples, so my entire hydrogen isotope sample that was pooled, and then we randomly generated new samples out of all of my actual data. All right, so we pooled them all and randomly populated a new sample for 1895 and 1910, randomly populated a new sample for my 1963 to 1984, and randomly populated a current data set. We did this 1,000 times. We did this 1,000 times, and then we calculated the tolerance intervals for each randomly generated data set. Then we kind of looked at random centroid shifts. So we can create a distribution of centroids and look at how they change when we compare them between time periods. So this example, if we're comparing 1895 to 1910 to our current sample 2013 to 2014, we're looking at how the centroid will randomly shift just through random generation, and then comparing it to our actual observed shift. So what we're looking for, if the centroid is the black dot, right, maybe the average centroid shift is represented by the blue circle, right? On average, just through random generation, the centroid between 1895 and 1910 to 2013 and 2014 is on average going to shift somewhere around the blue circle. But maybe our observed shift is something much greater than that, right? So that's kind of how we got at our p-values. So these here are our p-values. We divided the number of randomly estimated shifts greater than the observed centroid shift. And so our p-values are here on the right. So we did this for 50 and 75%. You can see the p-values were significant when we're comparing 1895 and 1910 to our current reading set. It was also significant at the 75% level when we were looking at our intermediate set, 1963 to 1984 compared to now. You can also see that there were two p-values, 1963 to 1984 compared to now at the 50% level. The p-value was 0.07, so not quite significant, but low enough to kind of be interesting. Same thing at the 75% level when we're looking at 1895 to 1910 compared to now. So moving forward from here, right, we've seen some significant change, and we would really like to explore why these things have changed. So one of the things that we're looking at is possibly urbanization and land conversion along the southern boundary of the area of occupancy, right? We might be seeing habitat destruction through urbanization, land use conversion as things become more agricultural based. We could do this through satellite imagery and land use mapping. Also, along the northern boundary, we might be seeing some effects of climate change. I don't know why I automatically did this, but we might be seeing some effects of climate change. So we might be seeing some colonization along the northern boundary. Some of the ways we might like to look at that is by using some NDVI, also looking at some temperature analysis. But what does this all mean? It means that this is kind of a new way to assess anthropogenic response, as it kind of shows a large range shift over time. So it also is a really cool way to use museum specimens. There are so many collections throughout the country, and some of them aren't really being utilized, and this is a good way to utilize them. Right? It provides us with some really useful information that could be used to determine rate of change or how things might be changing over a period of time, which is really important when you're looking at trying to conserve a species, conserve habitat for a species. You want to know how they're changing so you can make sure that you are managing it appropriately. So moving forward, it's going to be really important to maybe refining this method and applying it to other neotropical species, both threatened and endangered, and we'd really like to try and broaden the scale. This was a very regional study. We were looking at birds in western Pennsylvania, so it'd be really great to expand the study moving forward. With that, there are a lot of people that helped with this project. People at Youngstown State, a whole bunch of people at Cartermill, Steve Rogers at the Carnegie Museum of Natural History, who helped me work with the study skins, and Kim Sparks, who did the analysis at Cornell, all my feathers. So I think I have lots of time for questions if you have any.
I just As of, excuse me, looking at the GNIP preset data set through time, that's been a much underutilized sort of data set in the sense of looking for temporal changes in the base map over the last 100 years. Well, actually, much less than that because we don't have data going back that far. We have about 50 years.
Yeah.
And the ENSO signature seems to be in there.
Yeah.
And so, you know, what I'm hinting at is, I think you're doing fantastic work. It'd be great to go beyond this now, because you want to expand this more continentally, to really understand where those high variance levels are in the GNIP base map.
Yeah, so the GNIP, you have to use the assumption that it's not fluctuating too much over time, but the data just really isn't there. There's just not enough GNIP data. So it'd be really great if we could have more places collecting isotope values from the environment, right? Because the base map is based on stations that are taking samples from rainwater or water in the environment, right?
Yeah, you're comparing your stuff to a 50-year average data set.
Yes. So, but, you know, it kind of operates on the assumption that that average isn't changing. And it's possible that it is. So it'd be really awesome to have more GNIP data, more isotope data from the environment in order to make sure that the assumption is correct. More studies like that as well, I agree. Other questions? Yeah?
You're going to extend to more taxing?
Yes.
In which case, what would be your target?
We haven't discussed it yet, but we are hoping to expand the study, putting some grants in to look at more species on a much larger scale. Multiple abandoned stations, multiple museum collections, multiple species. So hopefully in the future.
Have you looked at area of those intervals to see if you could get range constriction over time or anything along those lines?
We have not, but would be a good idea. Other questions?
Thank you.